DNA damage and repair. We have seen DNA replicates very faithfully. Then even when organism gets exposed to environmental conditions and also the endogenous causes are responsible for the damage of DNA. So this type of damage of DNA always take place in the living organism's body. So the details of how DNA gets damaged and how it gets repaired. So in this chapter we will have to think over all these things. So DNA repair is a collection of processes by which a cell identifies and corrects damage to DNA molecules that encode its genome. In human cells, both normal metabolic activities and environmental factors like UV light, radiation can cause DNA damage and results in as many as 1 million individual molecular lesions per cell per day. Human genome is under constant attack of endogenous and exogenous reactive chemical species. Variety of genotoxic agents can induce chemical transformation of nucleotides or can damage the phosphodiester backbone of DNA. The relationship between cellular DNA damage caused by endogenous and environmental genotoxic agents, the cellular response, and the development and prevention of human diseases and aging are the areas of great interest in the medical, biological, and chemical research communities. There are about thousands of DNA damage events per day. So per day suffered by approximately 1,000 cells within the human body. DNA damage associated with endogenous species is more extensive than damage caused by environmental factors. Among the endogenous species, that damage cellular DNA are reactive oxygen species and reactive nitrogen species. These reactive intermediates means reactive oxygen species and reactive nitrogenous species. Nitrogen species are produced under conditions of oxidative stress. Among the external causes of DNA damage are ionizing radiations, solar UV radiations, now we will see types of DNA damage. First type is monoduct DNA damage. Second is diaduct DNA damage. Third is oxidative DNA damage. Fourth is hydrolytic DNA damage. And fifth one is damage due to ultraviolet and other type of radiations. So first, monoduct DNA damage. DNA damage takes place in a single nitrogenous base, that is monoduct. This produces a severe effect even on the organism. Monoduct DNA damage occurs by following ways. First, deamination. Second, tautomerism. Third, base analogues. Fourth, modifications of base pairs. And fifth, methylating agents. So in monoduct DNA damage, first one is deamination. So in this process, nitrogenous acid can bring about changes in the structure of nitrogenous bases of DNA. Adenine on deamination in presence of nitric acid changes into hypoxanthine. Similarly, histosine is converted into uracil and guanine into xanthine. Second, tautomerism. Here, molecule is able to exist in more than one chemical form, tautomer. UV radiation energy play an important role to form tautomerism molecules. Unusual base pairing also cause changes in DNA. Now, base analogues. Base analogues are placed in place of normal base pair during DNA replication. And this damages DNA. Modification of base pair. This takes base due to some highly reactive chemical which may modify nitrogenous base or bases, due to which base pairing mistakes may occur. As a result, change in pH also occur in surrounding environment, and surrounding environment may see damaged DNA. Methylating agent. Addition of methyl group also causes DNA damage. So after monoduct DNA damage, diaduct DNA damage now. In diaduct type of DNA damage, the alterations occur in more than one nitrogenous base of DNA. That is gross rearrangement of DNA, and it is called as a diaduct DNA damage. For example, pyrimidine dimers are produced. See here, it is thymine dimer. When DNA gets exposed to ultraviolet radiation, this thing happen. A group of two linked identical pyrimidines constitutes a dimer, and the phenomenon of dimer formation is called as a dimerization. During span of lifetime of an organism, its cells are exposed to many mutagenic agents which cause damage to DNA. Due to action of mutagenic agents, pyrimidine dimers are formed in which two adjacent thymine bases are linked to constitute thymine dimer. That is like this TT. On the same strand, same polynucleotide strand. Similarly, adjacent cytosines are linked on the same strand and constitute a cytosine dimer. Such dimerization occur when the cells are irradiated with ultraviolet radiation. Now, after diaduct DNA damage, third type, oxidative DNA damage. It is the oxidation of specific bases. Eight hydroxy deoxyguanosine is the most common marker for the oxidative DNA damage and can be measured in virtually any species. It is formed mostly by chemical carcinogens. Then hydrolytic DNA damage. It involves deamination or the total removal of individual bases. Loss of DNA bases known as apirinic sites can be particularly mutagenic, and if left unpaired, they can inhibit transcription. Hydrolytic damage may result from biochemical reactions of various metabolites as well as overabundance of reactive oxygen species. And last type, ultraviolet and other type of radiation DNA damage. So due to ultraviolet, damage of DNA occur which involves a cut in one or both the strands. Double strand breaks are especially dangerous and can be mutagenic since they can potentially affect the expression of multiple genes. Ultraviolet induced damage can also result in the production of pyrimidine dimers, now causes of DNA damage. After looking towards types of DNA damage, causes of DNA damage. So DNA gets damaged because it is constantly under pressure, tension, and external environmental conditions even. DNA molecule then should be protected from physical and chemical damages. Also should be protected from UV radiations and harmful gases.